Hello my dear friends, I'm Dr. Vaishali Bharambe, Professor Anatomy. Last time we have completed fertilization. Embryo has started to travel through the fallopian tube, see, and has finally started the process of implantation. Here. What happens after that? What happens in the second week? Come, let's understand. So here is the blastocyst, outer cell mass, inner cell mass, and the blastocele, which now gets converted on the seventh day. The inner cell mass gets converted into epiblastic tall columnar cells and hypoblastic flattened cells. And this is the day when a bilaminar embryo gets formed. And now your embryo is consisting of an outer trophoblastic group of cells and embryoblast divided into epiblast and hypoblast. Okay, so now with this we are entering into the second week. Come. Further development of the zygote, second week of intrauterine life. So steps of what we will learn today consists of uterus and its reaction to the embryo, changes in the outer trophoblastic cells, changes in the embryoblastic cells. Three parts of what all happens in the second week. So here is the uterus endometrium. Here's the myometrium. You know that the uterus after the menstrual phase undergoes changes. Slowly the endometrium builds up. With ovulation, from proliferative phase, the uterus goes into a secretory phase when it's secreting a lot of nutritive material in case there is a fertilization. So, now what happens? This embryo or blastocyst gets attached to the endometrium, begins to give out Syncytiotrophoblast cells in an effort to enter inside the endometrium or to get implanted. What changes does the uterus undergo at this moment? The changes it undergoes are called as decidual reaction. All the cells in the uterus endometrium begin to become big, distended, full of glycogen and lipids. Okay, this is called as the decidual reaction. Okay, and the decidua is a reaction that takes place throughout the uterus. Entire uterus undergoes this celebratory moment when it's preparing itself to nourish the embryo. It's like, a moment of joy for the uterus. Finally, there is an embryo. So, this is called as decidua. When the baby will be delivered, this entire decidua, which is lining your endometrium, will get delivered with the baby. So, here's your embryo, the myometrium or the muscle of uterus, the endometrium. The changes are in the endometrium, so focus on that. The endometrium, and here is the uterine cavity. The endometrium gets divided into three parts. The part of endometrium near the embryonic pole of the baby is called Decidua basalis. Part of the embryo covering the abembryonic pole is Decidua capsularis. And part of the embryo lining rest of the uterus is Decidua Parietalis. So what are the three types of Deciduae? Decidua basalis, capsularis, Parietalis. So this is asked as a 2 marker. So I've given this to you. You can take a screenshot. Let's move on. Uterus has celebrated. Uterus is ready to enclose the embryo and take care of it. What is the embryo doing? Embryo is trying to create a placenta for itself. Trophoblastic changes. Okay. So we will now begin to understand trophoblastic changes in the embryo. We will be talking about primary villus formation. Come. After that, we'll talk about embryoblastic changes. If you're enjoying this lecture, do please like, share and subscribe to my channel and hit that bell icon. Do visit my website for detailed anatomy lectures, surgical anatomy videos, downloadable notes, MCQs and much more. The link to my website vbanatomy.com is given in the description section. So let's talk about changes in the trophoblast in the second week. So on the day 7, you saw how the embryo was trying to enter further inside the endometrium by giving rise to an invading mass of cells called Syncytiotrophoblast. There are so many cells that you can't see their cell wall. They are just multiple number of nuclei. Below them is a single layer of cytotrophoblastic cells. So what are syncytiotrophoblastic cells? Mass of cells, you can't see their cell membrane, look like a multinucleated sheet. Cytotrophoblast, mononucleated single layer of cells, giving rise to the syncytiotrophoblastic cells. Understand that, okay? So, next step is that the uterus is celebrating. Okay, its blood vessels are enlarging. Decidua Basalis is full of blood. Come embryo, I'll take care of you. I'll provide you with nutrition. What is the embryo doing? The Syncytiotrophoblast, which was present only at the embryonic pole, now covers the entire embryo, as you can see. Cytotrophoblast remains as a single layer. Of course, here is the epiblastic and the hypoblastic layer forming the bilaminar embryo. At this moment, we won't be talking about it. So, on the 8th day, what happens? The Syncytiotrophoblast begins to show gaps in it. These gaps are called as lacunae, random spaces within the Syncytiotrophoblast. Soon, these get organized in a radial manner. See? And there are gaps and there are pillars of Syncytiotrophoblast in between. These pillars of Syncytiotrophoblast, see those, are called as Trabeculae. So now what your placenta is like, it's like an old temple like this. There are pillars and there are spaces. The spaces are called lacunae. The pillars of Syncytiotrophoblast are called Trabeculae. Get this picture in your brain. So now what happens? The uterus is also celebrating. The blood vessels are dilating to form sinusoids. See here, large dilated blood vessels, sinusoids. Now what happens? Let's quickly revise once. So what happened? This was the embryo entering into the endometrium. Decidua Basalis celebrating formation of sinusoids. Syncytiotrophoblast enclosing the embryo now from all around. Cytotrophoblastic layer, single layer. Embryoblast divided into epiblast and hypoblast, right? Now what happens? Syncytiotrophoblast develops gaps in it, lacunae. They are radially oriented. Pillars of Syncytiotrophoblast in between are called Trabeculae. We were up till here. Now what happens? The Syncytiotrophoblast erodes into the maternal sinusoids. It literally tears into it. Sinusoids are containing blood, right? So, take a look at what happens. This is the moment uteroplacental circulation is established. So, see, these are maternal sinusoids. And can you see how there is a rupture of the sinusoids and blood has flooded into the lacunae? Can you see these are lacunae full of maternal blood? And this is how mother's blood begins to nourish the embryo first time. So now Cytotrophoblast says, why should I not join your party? Why should you be the ones having the most of the fun? So Cytotrophoblast begins to give out cells that enter into each Syncytiotrophoblastic pillar. See there. This is the pillar, that's Cytotrophoblast entering into it. Now if you take a section of this, it looks like this. A core of Cytotrophoblast, a surrounding of Syncytiotrophoblast, surrounded by what? Tell me. Surrounded by maternal blood in the lacuna. Correct? This is called as a primary villus. Villus is you remember villi in the gut? So villus is a finger-like projection. This is a primary finger-like projection made up of a core of Cytotrophoblast and a surrounding of Syncytiotrophoblast. At this moment, it is bathed by maternal blood, and therefore the lacuna is now called as intervillus space. There are so many villi. So this is an intervillus space. This is day 11 to 13 in the embryo's life. Cytotrophoblast says, I am not stopping here. Now that I have entered, I will keep on growing. So see how Cytotrophoblast grows and completely encloses the embryo in a shell called the Cytotrophoblastic shell. This helps to anchor the chorionic sac in the mother's endometrium. Okay, so that is important for us. So, changes in the trophoblast in the second week, penetration of blastocyst into endometrium, lacunar formation, trabecular formation, sinusoid formation. Lacuna fusing with sinusoids, establishment of uteroplacental circulation. Cytotrophoblast entering Syncytiotrophoblast to form primary villus. Intervillus spaces containing maternal blood. Formation of cytotrophoblastic shell. That's the entire changes in the trophoblast in the second week. Note, separate lectures have been created and released on placenta 1 and 2. Links are in the description section. If you're enjoying this lecture, do please like, share and subscribe to my channel and hit that bell icon. Do visit my website vbanatomy.com for more lectures, detailed videos, histology diagram videos, MCQs and so on. Link is in the description section. So what did we learn? Uterus celebrating decidual reaction. Trophoblast helping to form primary chorionic villus. What is the embryoblast doing? What is the change happening in the embryo itself? Okay, take a look. Changes in embryoblast in the second week. So here is your embryoblast made up of upper layer of columnar cells called epiblast, lower layer of flattened cells called hypoblast. You can see that very clearly in the diagram, correct? This is called as a bilaminar embryo and this cavity here is called as a blastocele cavity, right? So a bilaminar disc. By day 7, embryo has divided into epiblast, which is also called primary ectoderm, hypoblast, which is also called primary endoderm. Remember, we are going to go from primary to definitive one day, but at this moment, the word primary matters. Primary ectoderm is epiblast. Primary endoderm is hypoblast. These structures together, keep your hands one on top of the other. These structures together form a bilaminar disc. Bilaminar disc. So now what happens? Epiblast cells move away from the trophoblast and a cavity gets created above them. This cavity is called amniotic cavity. It contains the amniotic fluid. It is lined by a flattened layers of cells which are coming from epiblast called amniogenic cells. So amniotic cavity, amniotic fluid, amniogenic cells. So many new words. Let's ask ourselves a simple question. Who forms the floor of amniotic cavity? Epiblast cells. Who forms the roof? Amniogenic cells. What is the content of amniotic cavity? Amniotic fluid. Clear? It's quite simple if you simplify it. Right. And the junction of amniogenic cells with the epiblast is called amnioectodermal junction. We will need it way later in embryology. Please note it. Right. So revise quickly. Epiblast cells, amniogenic cells, amniotic cavity. Right. Hypoblast says, why should I not enjoy myself too? So here is your hypoblast. Okay. You can see that it has given rise to a layer of cells that comes to line the blastocele. Okay. This layer of cells is called Husser's membrane. And this cavity now is called primary yolk sac. So primary yolk sac, roof formed by flattened cells of the hypoblast. It is lined by Husser's membrane. Okay. So now we've got two cavities, amniotic cavity, primary yolk sac. But this structure now moves away from the trophoblast by formation of a new layer called the extraembryonic mesoderm. So a layer develops between the trophoblast and the amniotic cavity and yolk sac. This layer is called extraembryonic mesoderm. In this extraembryonic mesoderm, soon a lot of cavities develop. These cavities coalesce together and they form one single cavity called the extraembryonic coelom or the chorionic cavity. What is it called? The chorionic cavity. Okay. So, this chorionic cavity now divides your extraembryonic mesoderm into two layers. An inner layer which lines the yolk sac and an outer layer which lines the trophoblast itself. Correct? You can see that here. This is the inner layer called Splanchnopluric Extraembryonic Mesoderm. Splanchnopluric Extraembryonic Mesoderm. And outside here, Somatopleuric Extraembryonic Mesoderm. Outer layer, Somatopleuric Extraembryonic Mesoderm. So let's understand the extent. Okay. Splanchnopluric lines the yolk sac, the primary yolk sac. Okay. So take a look. I've drawn a dotted line there for you. That's splanchnopluric. Let's take a look at somatopleuric. Somatopleuric layer lines the trophoblast, but it also goes on to line the amniotic cavity. Can you see that? That is the somatopleuric extraembryonic mesoderm, which actually contributes to formation of the chorion and the chorionic villi as well later. Revise: amniotic cavity, yolk sac. Chorionic cavity. This is the splanchnopluric extraembryonic mesoderm. This is the somatopleuric extraembryonic mesoderm lining the trophoblast and the amniotic cavity. There. Meanwhile, what you see is a portion of this extraembryonic coelom has not extended upwards. Can you see that it has reached up till here, but it has not extended completely. So there is a part of the embryo still attached to the trophoblast. This extraembryonic mesoderm is going to develop to form a connecting stalk, which is your future umbilical cord. Isn't it beautiful? I hope you're enjoying this as much as I am. Okay, so that's your connecting stalk, amniotic cavity, primary yolk sac. This connecting stalk, which is your future umbilical cord, is literally suspending the embryo in the chorionic cavity. You can see that. See, this is the embryo suspended in the chorionic cavity. Let's understand a few terms. There is a term chorion, there is a term amnion, which you will be asked very often. So understand it. The somatopleuric extraembryonic mesoderm along with overlying trophoblast form what is called as chorion. And this layer, okay, so therefore villi from it are called chorionic villi. The amniogenic cells form what is called as amnion. There is an amnion, there is a chorion. The amniogenic cells form amnion. They form the innermost membrane that encloses the embryo. So, this is your chorion, somatopleuric extraembryonic mesoderm plus trophoblast. This is your amnion, amniogenic cells. Villi from the chorion are called chorionic villi. Finally, we come to formation of secondary yolk sac. What happens is, a bit of the original yolk sac gets cut off, forming, see there, it gets cut off and it forms what is called exocoelomic cyst. And the actual yolk sac cavity becomes much smaller. This smaller yolk sac cavity is called as the secondary yolk sac. One last event before we finish off the second week is that embryo still doesn't know where is my head going to form, where is my tail end going to form. Embryo doesn't know. Isn't it time it found out? Come. So, we develop something called as a prechordal plate. This is the embryo, okay? Upper layer of epiblastic cells, simple columnar. Lower layer of hypoblastic cells, simple cuboidal flattened cells. One day, on the last day of the second week, the cells of hypoblast at one end become columnar and they get very tightly attached to the epiblastic cells. This region is called as the prechordal plate. It forms at the cranial end of the embryo. Okay, and that's when the embryo finds out that this is my cranial end and this is my caudal end, and therefore the axis of the embryo is also established. Okay, that's how we finish off the second week. Baby now knows where my head will form and where my tail end will form. I think that's good enough for a little baby to find out. So changes in the second week were formation of amniotic cavity, Users membrane and primary yolk sac, extraembryonic mesoderm, extraembryonic coelom, somato and splanchnopleuric extraembryonic mesoderm, formation of connecting stalk suspending the embryo in the chorionic cavity, defining chorion, defining amnion, formation of secondary yolk sac, and finally prechordal plate. Okay. So this was about the second week of intrauterine life. We also have lecture on third week of intrauterine life. There are 2 lectures. One is for gastrulation and one is on notochord formation. Okay, do watch both. Link to these in the description section. We have a lot of lectures on embryology, spermatogenesis, oogenesis, fertilization, first week, placenta, everything. Okay, do watch and learn. Please, students, share your doubts with me. I am here to make sure there is optimum education of anatomy available for students worldwide. With that comes my availability for you to clarify your doubts. Come put your doubts in the comment section and I will revert back to you. But do study, do understand. It's a journey of a tiny embryo. It's beautiful. We are on a journey. We are literally holding hands of an embryo and allowing it to grow, watching it grow. It's a privilege. Okay, I hope you enjoy this journey with me as much as I am enjoying it. If you're enjoying my lectures, do please like, share and subscribe to my channel and hit that bell icon. For detailed embryology lectures, anatomy lectures, surgical anatomy videos, do visit my website vbanatomy.com. Link is in the description section. I loved taking this lecture. I hope you enjoyed it too. Thank you.